Let's take a closer look at a chloroplast, the organelle where photosynthesis takes place. Photosynthesis consists of two sets of chemical reactions, the light reactions and the Calvin cycle. They occur in different regions of the chloroplasts. Chloroplasts contain stacks of membrane-lined discs called thylakoids, surrounded by a watery clear fluid called stroma. The light reactions are carried out by molecules in the thylakoid membranes, and the Calvin cycle reactions by molecules in the stroma. Let's explore these regions and their functions in more detail. In the thylakoid membranes, the light reactions transform light energy to chemical energy. Light energy drives the formation of ATP molecules from ADP and of NADPH molecules from NADP+ and electrons. Along the way, water molecules are split and oxygen is formed, which can be released into the atmosphere. In the stroma, the Calvin cycle reactions use the chemical energy of ATP and NADPH to combine carbon dioxide from the air with organic molecules to form new molecules like the sugar G3P. ADP and NADP+ are recycled and may be used again in the light reactions. A plant increases its biomass through the formation of these new organic molecules.
Next topic, chloroplast transformation. What is chloroplast transformation? Current achievement of chloroplast transformation. Chloroplast transformation, multi-step processes are involved to achieve chloroplast transformation. Specific species, specific and heterogeneous chloroplast transformation vectors are developed in a manner that franks the foreign genes and insert the heterologous recombination at predetermined and precise location in the plastome. So we can use another molecular biology techniques plus chloroplast transformation from one species to the another species for the development of different types of desired, hybrid, and also the different types of species. General three key conditions have to be fulfilled to achieve plastid transformation or chloroplast transformation. A robust method of DNA delivery into the chloroplast, the presence of active homologous recombination machinery in the plastid, and finally, the availability of highly efficient selection and regeneration protocol for transplastomic cells. Current achievement of chloroplast transformation in plants. How we can get benefits or achievement from chloroplast transformation in plants. First, chloroplast play a great role for sustained well-being of life on planets. They have a power and raw materials that can be used as a sophisticated biological factories. They are rich in energy as they have a lot of pigments, protein complexes capable of collecting sunlight in sugar produced by biosynthesis and in minerals imported from the plant cell. We know in the photosynthesis, carbon dioxide and water is transferred to the oxygen and carbohydrate by the use of chlorophyll for the production of biochemical energy and carbohydrate. Chloroplast genome transformation offers multiple advantages of a nuclear genome, which among other include integration of transgene via homologous recombinations that are able to eliminate gene silencing and position effect. Higher level of transgene expression resulting into higher accumulations of foreign proteins and finally significant reduction of environmental dispersion of the transgene due to material inheritance, which help to minimize the major critique of plant genetic engineering. Chloroplast genetic engineering has made fruitful progresses in the development of plants' resistance to various stress, phytoremediation of toxic metals, production of vaccine, antigens, pharmaceuticals, biofuels, biomaterials, and industrial enzymes. So, you read current achievement of chloroplast transformation in plants, just easy 3 to 4 points for your examination. Importance of chloroplast transformation. Another such importances that we found from the chloroplast transformation in plants. First, risks of transgene escape. Risks of transgene escape. Second, expression level, homologous recombination, gene silencing, position effect, disulfide bond formation, multiple gene expression, expression of edible vaccine, use of codon, expression of toxic protein. You just read the main points of the topic